Hello, Georgia Tech.
Woo!
This is TWiM, This Week in Microbiology, episode 208, recorded on October 16th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Today we are recording in Atlanta. We are at Georgia Tech, and we are guests of the Center for Microbial Dynamics and Infection. And joining me today, our usual TWiM co-hosts, Michelle Swanson.
Hello, what a pleasure to be here.
Thanks for joining us. And Michael Schmidt.
Thanks, everyone. Glad to be here in the beautiful state of Georgia.
Not far for you to come, right?
No, was not far. I just live across the state line in South Carolina in beautiful Charleston. Come visit.
And we have, for your listening pleasure today, we have a few PIs and their mentees. And let's start here on my right. We have video. If you're listening, you could pull over to the side of the road and look at the video. Here on my right, Frank Stewart. Welcome to TWiM.
Thank you. Hi, everyone.
To his right, Mark Hay.
Thank you.
Welcome.
Good to be here.
And next to Mark, Deana Beatty.
Yes. Good to be here.
And you were a former student with these two individuals,
Yes,
right?
that's correct.
So you needed two people to
I did.
keep you in line.
Keep up with all her great ideas.
And now you're a postdoc at UC
UC
Davis.
Yeah, with
So you
Jay Stocklewitz.
And you came back just to do this.
I did.
Very nice. Okay, then on the other side of the table, we have Marvin Whiteley.
Thank
Welcome.
you. Good to be here.
Who's instrumental in getting us here, right? And your postdoc, Gina Lewin.
Hi, really
Welcome.
excited to be here.
Welcome to the show. So we have two groups. We're going to talk about their two papers that they've recently published, which are on very different subjects, but really interesting and kind of illustrate the kind of science that's going on here. Before we get going, if you like our work on TWiM, please consider supporting us financially. We all do this on our own. We get very little support from others except our listeners. You can go to microbe.tv/contribute and learn about how you can do that. I'll tell you again at the end in case you forget. So let's first explore your training, all of you. I'm interested in where you're from and where you went to school. Went to school, that's right. That's a good way to say it. Let's start with you, Frank. Where are you from?
Sure. Well, I grew up in Reno, Nevada, actually.
Wow, I was just there.
Did you lose any money?
I didn't know people grew up there.
Yeah, right. Yeah, divorces and weddings. Yeah, I guess. So I grew up in Reno. I did my undergraduate in biology at this little school called Middlebury College in Vermont. Mm-hmm. Then I went back to Reno for 2 years and did a master's working on the microbiology of sea ice in Antarctica, and made a bit of a departure from that and worked for my PhD at Harvard on deep sea microbes at hydrothermal vents. And after that, a 2-year postdoc at MIT before coming to Georgia Tech in 2011. And I've been in the School of Biological Sciences since then.
So can you remember what got you interested in science in the beginning?
Yeah, absolutely. I think I can tie it all back to just loving being outdoors and having some exposure to nature. And I remember getting into the marine side of things via our family trips to the Oregon coast and exploring tide pools early on in life. I didn't really get into the microbiology side of things until my undergraduate, and my first research experience was looking at microbes in association with bryozoans up in the Arctic Ocean. And so that was, I guess, my first real foray into microbes. And I think I've stayed there since.
You've come a long way since Reno.
Yes. Yeah, absolutely. I still get back there, though. I try to.
So I visited the University of Nevada, and They have actually a quite vibrant research community there.
They do, yeah. So I actually worked in a research branch of the institution called the Desert Research Institute, which does quite a bit of work on atmospheric science and polar science. And that's where I got into the exploration of polar microbes and psychrophiles.
Well, if they got rid of the casinos, it would be a nice place.
It's a beautiful place, yeah, regardless of the casinos. Yeah.
But they're building Teslas there now.
They're building Teslas there, yeah. Right. And yeah.
Okay, Frank, where are you from?
Mark.
Mark, sorry.
There you go. Okay, you've got a cheat sheet. You should
It's wrong. That's the problem. That's the problem with cheat sheets.
Yeah. I grew up in central Kentucky in the Bluegrass region. I went to college at University of Kentucky and majored in zoology and philosophy. And then I did a PhD at University of California, Irvine. During that, I did all of my work on coral reefs and stuff in Panama through the Smithsonian Tropical Research Institute. Once I finished at Irvine, I did a postdoc at the Natural History Museum, the Smithsonian in D.C. Then I went to the College of the Virgin Islands in St. Thomas for 8 months because I wanted to be at a university in the tropics. And after 8 months on a small island, I decided I needed a continent, not an island. And so came back to the U.S. and was at UNC Chapel Hill for 17 years. And then moved here to Georgia Tech 20 years ago. And have been very happy with that.
So can you remember when you got excited about science first?
Probably at 2. I think I describe myself still now doing the same thing I did when I was a kid. I would, turn over rocks in the stream, catch butterflies, etc., and see who ate who and how the world worked. And that's still exactly what I do. I get paid better.
I can't imagine you remember 2 years of age, really?
I'm not sure, too, but certainly from 4,
Okay.
I was out running around in fields and streams and things, yeah.
Deana, where are you from?
I'm from Florida, near Tampa, and I did my undergraduate at Northeastern University in Boston. And after that, I went and worked at Scripps in San Diego at UCSD with natural products chemist Bill Finnicle and marine microbiologist Paul Jensen. And I worked there for 3 years and then came out here to do grad school at Georgia Tech.
And what got you interested in science? Do you remember?
I think it's pretty similar to Mark and Frank. Just as a kid, my parents always took us on adventures. We went hiking. We're always outdoors. Growing up near the ocean and just feeling at home when I'm near the ocean.
Okay. So you also found butterflies under rocks, right?
Possibly. I don't know if I have memories that vivid at 2.
Okay.
Dead butterflies.
Gina, where are you from?
I grew up in central New Jersey, and then
We're not going to hear yays for New Jersey?
No, apparently not.
And then I went all the way across the country to California to Pomona College for my undergraduate studies. And I really liked research that I was exposed to as an undergraduate, so I decided to pursue a PhD at University of Wisconsin-Madison. And I was in the lab of Cameron Curry studying insect-associated microbes and how they break down plant biomass. And then after I graduated with my PhD, I came and joined Marvin's lab, first at UT Austin and then now here at Georgia Tech.
How important do you think undergraduate research is to helping you make your career choices?
I think I really didn't, even with my limited undergraduate research, I had no idea what graduate school was like, but I don't think I would have felt very confident about applying to graduate school right out of undergraduate without doing undergraduate research. And I did a summer REU at University of Wisconsin-Madison as an undergraduate, which one showed me how awesome Madison is, and then also encouraged me to do research and showed me what research is like at a larger university.
Marvin, where are you from?
I'm from Belton, Texas, so a little small town about 60 miles north of Austin. So I think there were 10,000 people when I grew up there, and there's about 9,000 people now. So, and I graduated high school from Belton. I went to UT Austin for undergraduate. I was an aerospace engineering student for 2.5 years. And I was taking a Fortran programming class. That makes me sound really old. I'm not as old as I look, but and I guess the professor of the class came in and he was very upset. You clearly, like, a grant didn't go through. Something bad had happened that day, and he came in and he's like, I don't know why any of you are aerospace engineers. You're never going to get a job. The U.S. is never going to build planes anymore. You should just quit, be mechanical engineers. It's easier. And all of us were looking at each other, like, because we'd been through differential equations, you know, like these terrible classes. And I went across the street and started taking zoology classes. And so I changed my major, graduated zoology, and had gotten into grad school to go study birds and bird behavior, and got engaged. And my wife, I decided not to leave and sort of stay with her. She was doing a master's degree down at Texas State University in education. I went down there and met a guy named Bob McLean. I think maybe a few of you know Bob.
Oh, we know Bob.
Yeah, and so and did a master's degree with him. It was the first time I ever did microbiology, and and he sort of helped me kind of think about what I want to do in life. And he cold called Pete Greenberg one day at Iowa and said, I have this guy that you have to fly up and meet. And I went to Iowa and got a PhD with Pete, and then did a short postdoc at Stanford. And then I was at Austin for 11 years, and then moved to Georgia Tech 2 years ago as faculty.
So someone commented recently on one of our road episodes, these people keep dropping names left and right. I wish I could go to just a few of those institutions. It's really remarkable when you hear where everyone has been, right?
Yeah.
All over the place. So science is a mobile profession, and you can go to pretty cool places as well. So that's why I like to hear where people have been. Okay, let's talk about your science. We have 2 papers. Let's first start with Gina's, which was published early this year, I guess, right?
September.
Last year? This year?
2019.
That's still
Last
there
month.
this year. Okay, large-scale identification of pathogen essential genes during co-infection with sympatric and allopatric microbes. Not an elevator-friendly title, is it?
No.
So if you were in an elevator and someone said, what do you do? How would you explain that to them?
I usually tell people that I study microbes that live in our mouths, and I'm interested in how they interact with each other and why those interactions matter. But then this paper was expanding on just from studying microbes that live in our mouths, but also comparing to interactions with microbes that don't live in our mouths.
So would you explain the sympatric and allopatric parts, or just leave that?
When I talk to somebody in the elevator?
Yeah.
Probably sometimes, depends on how interested they are.
So if I understand this, this paper is about genes that an organism needs that differ when they're alone or with other organisms. Is that fair?
Yeah.
And did we know that this was an issue before you started this work?
Yeah, so Carolyn Iberson, who's a postdoc in our lab, published a paper in 2017 looking at this with Staphylococcus aureus, thinking about this question of how do these essential genes, these genes that an organism needs to survive, change in monoculture versus in co-culture. And she saw that there were differences, and in general, the presence of another organism made staph require a lot more genes. So, and then since then, a little bit more has been done looking at essential genes during mono and co-culture and other groups. But what we really wanted to do is ask that on a much larger scale across a much more diverse group of microbes.
So I'll have you pronounce the microbe once only so that everyone knows what it is.
Well, I'll say what I say, but then other people have other interpretations of how it's said. I call it Aggregatibacter actinomyces accomitans.
Okay.
Everybody clap.
So we'll call it AA, right?
Yes.
That's what everybody calls it.
That's the pet name, AA.
Well, as Michael has pointed out, has other meanings as well, so you have to watch out. But AA, so I'd like to hear why AA and why the particular animal model and the other microbes that you mixed with it? Why did you choose all of those?
So I'll start with maybe back up even a step from there and talk about why the oral microbiome is a good model for studying microbial interactions. A lot of work that we hear about and a lot of cool things that are being done with microbiomes is in the gut. But really, the oral microbiome, we know a lot about what's there. Most of the microbes are culturable. People have been studying the oral microbiome for so long, it's easy to sample. And people have been studying not only who's there, but also the interactions. And there's even been really great work done by Jessica Mark Welch and Gary Borsey looking at the spatial structure of plaque. So it's a really good system for studying microbial interactions, in addition to it being really important for human health between cavities and gum disease.
And you can do these experiments at home. You just simply don't brush your teeth for a couple of days and you feel AA literally welling up at your gum line, and it's doing all of these remarkable behaviors that Gina and Marvin are going to tell us
Maybe
about.
you do, Michael.
I brush regularly and floss.
Yeah, I definitely brush my teeth better after starting to study oral microbes.
But not only that, our mouth is the front door to the rest of our GI tract. And so if you think about all the foods we eat, we're probably constantly introducing invaders or guests.
Or disturbances.
Yeah.
Yeah.
I think you're also, I mean, one of the things most people don't know about the oral cavity is every time you brush your teeth, you actually introduce these microbes into your bloodstream, and they can be cultured out of your bloodstream every time you brush your teeth. And so they've been implicated in a lot of everything from endocarditis to a lot of different diseases because they have ready access to the bloodstream. So.
Yeah, you're bacteremic 4 hours after you brush your teeth. So if you're the average individual who brushes their teeth twice each day, you're bacteremic for 8 hours.
Sounds like a good reason not to brush your teeth.
No, it's your body has adapted to the presence of this, event.
My dentist, who I love, after I get a teeth cleaning, the dentist comes and does various things, including checks my glands, and they're always swollen. And I said, yeah, but you didn't check them before. She was in there cleaning and grinding away. It's my neutrophils have come screaming in.
Oh, they have.
No, you wanted to control.
It is a really cool system. Our undergraduates here at Georgia Tech run a semester-long lab as part of the intro microbiology class that focuses entirely on the oral microbiome. So the first half of the semester is they design some experiment that tests the effects of a perturbation to the oral microbiome, and then we explore it by examining how the community composition changes. We don't look at how the abundance of the microbes change, but they design some pretty cool experiments. They explore things like the effect of brushing, what happens 5 minutes afterwards or 10 minutes after applying mouthwash, and the effects are pretty dramatic. The community shifts. It shifts back, but it shifts pretty quickly.
I think it's interesting. I mean, there's a lot of experiments you can do in the mouth that you can't do in the gut. I mean, you just can't sample it in the same kinds of ways. And I think
In the same time frame.
And the same time frame, you can do temporal studies in the mouth.
So that leads to my question. If the mouth is so cool, why did you put these into the thigh of a mouse?
Here you go.
Yeah. So there's a few different reasons why we do that. One is it's really hard we wanted to do this experiment in the presence of an immune system of a host, because we know that's going to be influencing the interactions between these organisms. It's really hard to do oral animal studies. So you can, as you said, it's the opening to everything. And so the thigh, these abscesses have become a pretty common model for studying oral microbes. And AA causes abscesses naturally in humans when it gets in our bloodstream. It can go and cause abscesses across the body, in the brain, and in the mouth. And a lot of those are, we think, co-infection abscesses. There was just a paper that came out from Iowa about abscesses between AA and Actinomyces israeli. That was one of the organisms we used in our paper, and there's other reports of that as well.
Okay, so now can you describe this experimental system? What actually did you do?
Okay, so we are interested in what genes AA needs to survive when it's on its own compared to when it's with other organisms. And so we chose 25 microbes some that are from the oral cavity and some that are not from the oral cavity. And we did pairwise co-infections into the inner thigh of the mouse, creating abscesses. So each co-infection was just AA plus one of these other 25 microbes. And then we used a transposon sequencing library of AA for these experiments. And so that is basically a mutant library where we can ask at the end, using sequencing approaches, what genes are important in each of these different conditions.
So if you start with just unmodified bacteria, so you're going one-on-one, AA with another bacterium into the thigh, what do you find?
So if it's AA plus just one other microbe, we find a range of things depending on what the other microbe is. I think in most cases there was an abscess, but the size of the abscess could vary. And then we found that in some cases, AA was much more abundant in the abscess than in others. And in some cases, the other microbe was much more abundant than AA. And what was interesting was that we found that AA was generally more abundant with microbes that are not commonly found in the oral cavity. And I should mention that AA is really specific to the oral cavity. So it's really only found in the oral cavity of humans and old world primates. So it's not, environmental or also found in other places.
So what do you think about that observation, that it does better with other bacteria from the mouth? That's what you said, right?
It does better.
The other way.
side.
The other way.
It does better with microbes that are not from
Right. So what do you
the
think
mouth.
that means?
So I guess I've argued this a lot of different ways to myself. I think I initially thought that maybe organisms that are from the same environment, you think that they've evolved to survive in different niches, to use different nutrients. And so I thought that, that they would, all do better when they're together because they're going to kind of split up the environment. But also you could kind of make, I think what we're seeing is the opposite argument, that these organisms that are from different environments, they're really surviving differently, that there's a lot more competition between microbes that are all from the oral cavity versus organisms that are not.
The niche that they find themselves in the abscess is truly with the non-oral microbe. It's a new opportunity for them.
Yeah.
They're looking at it as a new opportunity, so they do the AA does much better. While if they're infected with their old friend and neighbor, they already know what to do with that friend and neighbor, so they know that they can't get too out of hand or their neighbor is going to play whack-a-mole and bring them down to size.
And maybe there's more competition. There's only so many nutrients that organisms in the mouth might be able to use, and so they're limited and competing more.
Okay, then next you had this library of transposon insertions,
Yes.
right? Which means you disrupt many different genes.
Mm-hmm.
A single gene in each each bacterium.
And these are able to grow in broth, right? So
Yeah.
you don't see a lot of the insertions because they're lethal.
Yep.
Yeah, so there's a number that you don't see even in rich media in the lab because they're always important for growth. So we kind of exclude all those from all of our analyses.
Right, and then you do co-infections with those in the thigh of the mouse again, right?
Yeah, so actually all the co-infections were with that, but we just analyzed it for counts and then for the library, but yes.
Okay, so then when you do that, you do also one-on-one?
Yep, it's all
AA
pairwise.
with one, pairwise.
Yes.
So what do you find then?
So we find that, so there's a set of genes that A needs in mono-infection when it's on its own. There are about 200 genes, I think, that AA needed. And then in addition to that, there were 593 genes that were required with at least one of the co-infection species. So
about a third of the genome is essential with a partner, with at least one partner.
But in broth, you don't need them.
These are all genes that are not required in broth, but are now required in the abscess.
So are they required always with a competitor, or just in the abscess alone?
So what was interesting was that of the genes that were required in monoinfection, they were all, I think they were all also required in co-infection. So they weren't always required in co-infection, but there was some other microbe, in the presence of some other microbe, A always required those genes.
Right.
I think there's something important sort of to mention here. I think Carolyn had seen this in her paper, was that something that I think most people may not have expected. When you put bacteria together and you ask what genes you need to live, what happens when you add another bacterium, you get a lot more genes that AA or staph requires if another bacterium's around. The number of genes you need to survive in an animal goes up. But it actually turns out that there's a lot of genes that you might need by yourself. You put another bacterium in, you don't need those genes anymore. And I think this is sort of one of the biggest, I think, most important aspects, I think, of both of these papers was that a lot of people, if you go in and you look for, say, therapeutic targets, right? They do T-and-seq and you put it in an animal and say, oh, these genes are required. These are good therapeutic targets. But it actually turns out if you put other bugs in, a lot of times those genes are not required, right? And we know that almost all these infections are polymicrobial. There's multiple bugs. And so you have to really think, when you're thinking about an essential gene and something you need to survive, you got to think more broadly, and it's not a single bug. And I think that's something we're probably not doing. And I think that's the thing that's coming out.
Yeah, the way I thought about it in reading this beautiful work is that you might be able to scavenge crumbs from a sloppy neighbor, but other neighbors might be more stingy and they might leave fewer crumbs. And so what I need will depend on what my neighbor is leaving.
But we did identify a set of about 60 genes that A always needed, whether it was in monoinfection or with any of these other microbes.
In the thigh, right?
In the thigh. And so those genes, a lot of them were involved in respiration. So A can respire or ferment. And so it implied that no matter what other microbes were there, or if no other microbes were there, respiration was really important for A to survive in the abscess.
Mark, you were going to say something?
I was going to say and I'm more of an ecologist with larger critters than I am a microbiologist, but I think Marvin's comment is something we see over and over in diverse communities, is if you understand the physiology, it often doesn't predict much at all about what happens in the real world. It sets the limits on where you can be, but competition and predation and parasitism and commensalism sort of overwhelms that quite often.
So the other part I found really cool are these differentially required genes, which you call code, right? Mm-hmm. They're needed, depending on whether they're mono or co, they may or may not be needed, changes, right? So tell us about those.
So this term code gene, these conditionally dependent essential genes, Carolyn described those first in her paper. So these are things so there's two types. There's there's genes that are required in monoinfection and then alleviated in essentiality by co-infection, or they're not required in monoinfection and become essential in co-infection. And we saw that some of the co-infections in my study, there were a lot more genes that were alleviated. And in other co-infections, a lot of genes became essential that were not essential in monoinfection. And it was interesting, the three conditions where there were just a large number of essential genes alleviated relative to monoinfection were these non-oral co-infections with non-oral microbes. So something about the presence of those organisms is alleviating a lot of the genetic requirements for AA in the abscess.
I think the way I've always looked at it, I thought it was, I mean, I don't know that we expected this. It was kind of interesting, though, that it's a bug AA has never seen, but AA needs fewer functions when that bug's around, right? And so what we sort of think about this is when you've co-evolved with something, you come up with these very specific interactions, right, where you've evolved. These other bugs may obviously don't know about these interactions. They're just dumping all kinds of things out into nature, I think. These are bugs with really big genomes, make a lot of amino acids. They do a lot of things, and AA just sits there and gobbles all of this stuff up, right? And there's never been sort of any coevolution for these sort of these trade-offs. And so I thought it was kind of interesting that was.
I mean, also, as you said before, since the mouth is bringing in microbes all the time, so maybe it's not unreasonable to expect that AA will react to microbes it's never seen before, right?
So let me ask the flip question. What if you did this with the non-oral species first, and then ask what do the oral microbes do? And you monitor the genome of the non-oral species and ask, do they have these co-genes that enable them to have a selective advantage if you were able to come up with an abscess model in the oral cavity?
Yeah, I think it would be really interesting because you're putting that non-oral microbe into a foreign
Different environment.
And so
Do you think the reciprocal will hold true?
I think there's a good chance. I guess my intuition is that in that environment, the organism would need fewer genes because it doesn't have a lot of genes that are specifically important to survive in that environment. So it's kind of a similar balance to what we're seeing with AA.
I think the coolest thing is the microbe that alleviated the most genes was Saccharomyces, though, right? So I mean, this is
fungus.
Yeah, right. I mean, which was I think pretty interesting, right? That we didn't even know if it would survive in a mouse, right, at those temperatures. Right? I mean, we didn't know, and obviously did. So yeah.
Did you try Candida?
Yeah, we have Candida as well, and Candida also alleviated a large number of genes.
So that's one you might find in the mouth?
Yes.
Right?
Absolutely.
All right.
Tell me about the conversation where the two of you decided that you were going to use 25 different partners, like not 4, not 7, but 25.
That's
It wasn't much of a
25.
conversation. It was just a blank look when I said we need to do 20 or 30.
Yeah. Well, and this project was started by the second author on the paper, Apollo, before I joined the lab. So I'm really grateful to him because he was the one who went out and found all these microbes across labs at University of Texas and from across the country, and he did the initial infection. So he figured out how to grow all these different microbes and get them into the mice. And so, yeah, he had a huge and really important role in this paper, and I'm really indebted to him for doing that.
So I've got to ask Dr. Whiteley, why 25?
I mean, I wanted 50, but I mean,
Why? What was the rationale?
Well, I mean, what I was trying to do was explore metabolic space, right? I mean, that's obviously what you want to do, right? You want to go from bugs that are highly specialized bugs with maybe a couple of thousand genes to bugs that have 6 or 7, 8,000 genes. And then because what I was trying to do was functionally tease this apart, right? Like, so if you put a bug in with more functions, how does it affect? And that's exactly what Gina saw. She saw a correlation with function.
So you wanted to kind of saturate the landscape, and that required using more.
Yeah, that's
cover
right.
That metabolic.
Yeah, and I don't have to do the experiments. I mean, so I mean, it's, you just go do it, right? I mean.
Well, do you think you've saturated the landscape?
Well, Gina actually has. Well, you can tell them you have a figure in here about what you might find if we added more bugs.
Yeah. So we had data from 25 microbes in this paper, but then we asked, well, and we found 593 essential genes across all those conditions. But what if we added a 26th pairwise condition, a 27th, a 28th? So I modeled that out, and I predicted that there was about 800 genes within the A genome that would be required in some co-infection. And in context, A has a pretty small genome. It's like 2,400 genes. So that's about a third of the genome that we think are important for mediating these co-infections.
If you had infinite co-infections, essentially, it was modeling if you had infinite co-infections.
So we've saturated a fair amount of that space just in the study, but there's probably things that we haven't seen yet.
And we know that most abscess infections are polymicrobial, so that makes sense, doesn't it?
Absolutely.
That a lot of the genome would have to be dedicated to competing in that space.
So earlier you mentioned that one of the advantages of using a mouse is you have an immune system.
Yeah.
Do you ever think about getting rid of it and seeing what happens in co-infection?
Yeah, we've played with those ideas. We have not done any of those experiments yet.
I suggested that a couple of years ago, but she would have to do all of this again. And so.
Yeah.
in a mouse model,
Well, you have to decide which
I
mouse
think
to use.
kind of pulling apart the role of the host is a really interesting component of this project, how much of these interactions are direct versus how much of them are indirect and mediated through the host. We've also talked about just redoing some of these interactions in vitro to get a sense of how much of the interaction is coming from the host. But it's also hard to model the nutritional environment in the abscess, so it would be
plus
complicated.
the immune system coming in and what the immune cells are signaling the microbes to do and vice versa.
Yeah, it gets complex pretty quickly.
I think the way we'll probably go is look on, the Gina's just learning about this, but the coinfections that have the most essential genes and the least, you could go into a skid mouse or something, right, and then ask how did those change?
How did the immune
You don't have to do
system
3
react?
or 4, yeah, and so was it a direct interaction between the bugs? Are you impacting the host, which is then impacting, the essentiality of the pathogen? So.
Another thing I think would be interesting is these are acute infections that you did. Do a chronic infection model and see if things evolve with time, right?
Yeah.
If dependencies change over time.
Yeah.
Would that be interesting?
Yeah. Adding a time scale to this would be really interesting. I mean, there's so many variables because we are doing so many microbes at once, we couldn't really play with all these other things like time, like inoculation, abundance,
Inoculant size.
Yeah.
But I think we should, TNSeq provides you with a fossil record, though, right? Because you put all the mutants in, and then they either disappear or don't, and you assess later, right? You don't know exactly when they left, but they will. If you weren't fit early or late, you'll still come out in the end. And so I think that's the nice thing about the technique. Right. You get a fossil record.
Now, you could do this with any bacterium, right? It doesn't have to be AA. So you've established some principles here, but do you think you'd learn more by looking at other combinations? You don't have to do this, but just, what do you think?
Marvin may make her.
Good idea.
Is there something to learn from looking at other bacteria besides AA?
I mean, definitely.
Which one would you pick? Name your poison.
I think I would pick a more generalist
Like
microbe.
Fusobacterium nucleatum?
Or even a non-oral general, a microbe that you can find in a lot of different environments. So maybe something like Fuso or something that can grow more environmentally and in the host. And that can use a lot more carbon sources.
Pseudomonas.
Yeah. And we're interested in Pseudomonas and staph, and we've done a lot of these experiments with those bugs, also looking at the same kinds of things. So yeah.
But in terms of the oral cavity question, I think Fusobacterium, which is involved as an oral pathogen, it's another opportunist because when it's in the oral cavity, it's an opportunist. And when it ends up elsewhere, rotting dead bodies, it's an opportunist. So
Fusobacterium is actually really important in the mouth. I mean, us and Jorge Frias Lopez's lab and others have shown that when you transition from healthy to disease in your gums, Fusobacterium produces a lot of butyrate. And so you see this butyrate production, and it really transitions you to this more dysbiotic community. So it's a function that's actually conserved. You always see this function in humans.
Butyrate is so important to good gut health.
Yep, that's right.
And so there you want Fuso.
Yeah.
So can you share what you'd like to go next with this before you leave?
Yeah, so...
You're not ever leaving, are you?
Never.
It's a good answer.
So I think the part of the study where I'm thinking about starting with the follow-up is focusing really on these conditions where a lot of the genes that are required in mono-infection are no longer essential in co-infection, and understanding really why that is. But I'm also interested in taking what we learned here and building up to more complex communities. And I've been playing around with how these interactions are important for the stability of those communities as they get more complex and as you have transient organisms coming in.
Mm-hmm. What are you going to do next when you actually leave? 'Cause you will at some point.
That's a good question. So I'm interested in applying for faculty jobs, both probably at smaller liberal arts colleges and potentially elsewhere.
And you were impacted by your undergrad, right, for that decision, I think, at Pomona, I think,
Yeah.
So you would like to have a smaller institution, but still do research, right?
Yeah, I definitely would like to still do research. And I really, wherever I end up, I want to be somewhere I really, I got to do a lot of mentoring and teaching, especially as a PhD student, and I want to be somewhere where that's a really big emphasis.
Okay.
Can inspire the next generation.
Yeah.
That's great.
Yeah.
All right, we'll return to you at the end. I have a few more.
Okay.
But I want to hear about coral. So Deana, you published a paper this month. Science Advances Variable Effects of Local Management on Coral Defenses Against a Thermally Regulated Bleaching Pathogen. Maybe more elevator friendly than the other title.
Not by much.
But maybe it's important to set first why coral reefs are important.
Sure. They create habitat for a great wealth of biodiversity. They're often compared to tropical rainforest in terms of the number of species they support and also in terms of the productivity from those systems. So the corals are interesting in that some corals can live without a microalgal symbiont, and other corals require this algal symbiont for food. And so the symbiont performs photosynthesis and feeds the coral.
And so you make a point very early on in this paper that corals are declining. Mm-hmm. How do we know this?
Through observation.
We look and we go and see them and we see they're having problems. Mm-hmm. Is this everywhere or just in certain places?
I think it's variable and patchy, but the Caribbean declined pretty greatly in the 1980s due to a multitude of stressors. So there were disease that came through and wiped out a lot of the corals there, as well as hurricanes, overfishing, nutrients, pollution, and the Pacific has remained pretty healthy up until just the last couple of years. In 2016 and 2017, there were bleaching events in the Great Barrier Reef where we lost about 50% of the corals there.
Yeah, actually, they declined. We'd lost half of the corals in the Pacific over the last 30 years before that. Then the half that were remaining, you lost in 2016-17 in the Great Barrier Reef.
Is there any evidence that there were previous coral losses longer years ago that then were reversed?
Well, corals have gone extinct mostly extinct about 5 times in the fossil record and come back. So over the long term, yeah. But in the decades before the 80s, there's very little indication of broad-scale bleaching.
Okay.
Could you tell us just a bit more about bleaching? It's a term that we hear in the popular press a lot. Does that mean they've lost their symbiont and they're dead, or they need to regain?
So corals can live without their symbiont for a period of time via filter feeding, but if they're not able to recover those symbionts, then they will slowly starve to death.
Got it.
So there are also pathogens that cause bleaching, right?
That's correct.
Can you tell us about that?
Sure. So there's a couple pathogens, Vibrio chelonae and Vibrio coralliticus, and a few others that are closely related to those species of Vibrio that can go in and they produce toxins that are harmful for the microalgal partners. And so they will essentially poison the corals, they lose their symbionts, and if the temperatures are warm enough, they can cause tissue lysis and mortality.
And there's also a coral microbiome, right, that is important for the health.
Mm-hmm. Yes.
And what do we know about that?
We know a little bit about that. So the microbes in the microbiome can fix nitrogen and provide nitrogen as a supplementary source of nutrients. And they also can produce antibiotics that can fight off potentially harmful bacteria as well.
And so you mentioned that the decline of coral is associated with changes in the microbiome. Do we know if they're causative or just a consequence of?
It's tricky. In a lot of cases, I think we don't know if they're a cause or a consequence. There are just a few cases, though, where it's been shown that they can cause disease.
Okay. So now your study takes place at a coral reef in Fiji, right? Mm-hmm. Can you tell us a little bit about the study site and why it's useful for for looking at coral decline?
Sure, yeah. There's a fringing reef around the main island of Fiji, Viti Levu. So it's very accessible from shore. You can just swim right out and work. It's a shallow near-shore coral reef area. And the really great thing about working in Fiji is that the local communities there have established marine protected areas, and these are replicated along the coast. So they're just in front of each community where they really take an active role in monitoring the health of the reef and making sure that no one's going in and poaching or taking fish from that area. And so we have these really nice replicated areas of reef that are either protected, there's no human use, no fishing, and just adjacent areas that are used by those communities on a routine basis to harvest fish and other invertebrates as a primary source of food.
And over the decade or decade and a half that those have been in place, the non-fished areas have about 60% coral and virtually no seaweeds. And the fished area is almost all seaweeds and almost no coral. And so it's kind of like the Caribbean of the 1960s versus the Caribbean of today, but we've got them replicated and interspersed.
And it's Fiji.
And what?
And it's Fiji.
And
About his lab environment.
yeah, and that has both positives and negatives.
So why is fishing a problem for a reef?
There are a number of herbivorous fish that are really important. They play important functional roles in removing different species of macroalgae.
Okay.
And macroalgae can be very harmful for corals via shading, abrasion. Also, they can produce chemicals that are toxic to the corals.
Okay, so the Fijians are good about not fishing where they're not supposed to.
Correct.
Nobody violates it or not on a level enough to make a difference, right?
Depends on which villages you're working with, but the 3 we worked with, yes, very good. 2 in between, not so good.
So is this a area you've studied for many years?
16 years.
And how did you initially establish this collaboration, I guess?
We got a big grant to go to Fiji.
Before you got the grant, how did you?
We gave the Fijians lots of money. Just over time of working, we had a partner university there, University of the South Pacific, and they had good relations with various villages. But there's tremendous variance in what a marine protected area in Fiji means, just like there is here. And a lot of that has to do with the local governments. It's not something that the federal government does. It's something that each community does. And it's very localized. And at the villages where we were, if you go out and fish, there'll be a village meeting within the month, and everyone will shame you. And so people tend just from shame and sort of public pressure to do the right thing.
Pretty different from here, isn't it?
Yeah.
No public shaming here.
Mark, since I haven't been there myself, how large are the MPAs relative
These
to
things are small. They're about 0.4 to 0.8 hectares. So they're a few football fields in size. But literally when you swim out of the marine protected area into the fished area, you just hit a hedge of seaweeds right at the border of each of them.
So Deana, when you were looking for labs, was this an attraction, Fiji, or?
Mark came and gave a talk actually at Scripps, and I remember seeing some of the images, and of course it's very beautiful, but I was really interested in community ecology and the role that microbe-host interactions might play in scaling up to affect ecosystem health. So seeing his talk, that definitely sparked my interest, but it wasn't for another year or 2 before I thought about grad school.
Did Mark give a talk at ASM a few years ago? Was it you? No?
Ask Mark.
Not that I remember, but portions of my life I don't remember very well.
Okay. I thought someone gave a talk about the decline of reefs, showed incredible movies where the reefs are dead.
Yeah, you were sitting next to
Yeah.
Chris Kellogg.
Yeah.
Probably Forrest Rower.
No, it wasn't Forrest.
I remember.
It looked like you.
I don't remember.
Okay.
You don't remember who?
You have a doppelganger.
The doppelganger. Okay. Anyway, so tell us the study design. You obviously went to Fiji.
I
right?
did, yes.
It's a tough project.
Well, the airline
could be.
flight to Fiji is the tough project part.
It takes quite a while. I think it was about 30 hours from the time I left my apartment in Atlanta until I arrived at the house we worked
There's
at
multiple
there.
stops, I guess, right?
LAX is the main layover.
LAX and then
And straight into Fiji.
Fiji.
Yeah.
Okay.
Yeah.
How many times have you been?
Oh, goodness. I think probably 4 or 5 times.
You got a lot of miles, right?
Yeah, I guess so.
Okay. So what's the study design? What do you do?
Sure. So we selected a series of sites that are paired. So we have the protected area that's relatively healthy, lots of coral, lots of fish, very little macroalgae. Adjacent to those areas are fairly degraded reefs, very little coral, lots of macroalgae, not much fish. And so I went out with my lab mate, Cody Clements, and we swam around all of the areas that were fished areas of reef, trying to find candidate species that could be consistently found in those areas. Because coral cover is so low, it can be a little tricky to find those species. But once we did, we went to each of these sites and collected a small number of fragments of the coral. And a piece of the coral was preserved to do subsequent microbiome analyses via sequencing of the 16S gene. And a few fragments were used in what we're calling generating this coral wash or coral water.
Now, the species, you just look at it and you know what it is, right? Visual inspection initially. You have a sheet of photographs underwater with you, or you know what they look like?
Well, it's kind of passed down through the lab and people that are trained in identifying corals. And so that knowledge just gets transferred down.
And you're allowed to go out and you're snorkeling, I presume,
Yes.
right? Do you need a permit or anything? Or
We get an okay from the Fijian government to go do this, and then we have to go set in each village and drink kava and tell them what we're going to do. And I say I wear a dress and sit on the floor and do drugs with the chief. And then they eventually let you go do this.
That's why you can't remember parts of your life.
Yeah.
So then you break off bits. You put them in a Ziploc or something?
Containers that have been sterilized.
Containers. But how do you do that with you take it out of the water and then put it in on
No, you take the container underwater, you open it up so it gets a little bit of water in there, and then you move the fragments into the container.
All right, so you collect those. Now, do you do the analysis there, or do you bring everything back here?
Oh, all the samples came back here, and that was a very nerve-wracking trip, seeing my entire dissertation in 3 coolers getting checked into the Fiji Airways.
Because they don't always arrive.
Really?
You did a quick freeze too down there.
Just
right?
at minus
Preserve.
20. Yep.
And then it has to fly frozen on the plane, and
Yes.
you light a candle at the church before you go, and you light a candle here, and you pray.
Yes.
But you did this multiple times, each time collecting additional specimens, and they all became part of the study, right?
These were all collected in 2014 at the same time. So.
Okay. All right, so you bring them all back, and then let's talk about the wash, what you call the coral wash. What's that for?
So we wanted to obtain some material that would be representative of the coral halobiont. So there's a number of organisms that live with the coral. So the Symbiodinium is the microalgae that performs photosynthesis. There's viruses and bacteria, fungi, and collectively they're part of this meta-organism that could potentially have antibiotic effects toward this pathogen. So we wanted to obtain some sample of that. And from Kim Ritchie's work, she's on this manuscript as well. In 2006, she published a paper in MEPS where she was able to harvest mucus from Acropora palmata in the Caribbean and showed that this sterilized mucus had antibiotic effects towards a variety of environmental microbes, including Vibrias. So we wanted to know if we are able to reproduce this effect with Pacific corals and if protection status and reef state might influence susceptibility or resistance to this pathogen. But unfortunately, a couple of the species we selected did not readily produce mucus, and so that's where we came up with this other idea. I just looked around the lab and tried to find what I could, and we had some glass jars and we had seawater. We didn't have solvents, so I thought, well, I'll just shake it up in there and see what I get.
Seawater from the same area?
Yes.
Yeah. Okay.
And wasn't really sure if we would detect an effect, but we did. So that was really exciting.
You found an antimicrobial effect of the wash, right?
After it had been dried down and aliquoted into 96 well plates.
So what did that actually look like? What was the day in the lab where you saw, aha, there's an antimicrobial activity?
It's pretty exciting and surprising.
But what were you actually looking at or doing?
So the 96 well plates are read on a spectrophotometer, and then you can look at the density of bacterial cells in the treatment in the presence of the dried coral material or the absence of that.
Well, we were actually looking at the metabolic activity of those cells via...
Well, and later we added an indicator reagent so that we could see differences in the metabolic byproduct of tetrazolium chloride. But
Akin to what Biolog does. You were effectively doing the Biolog assay where you were watching how quickly they would consume a particular substrate.
Yes.
So did you find differences between the fished and non-fished corals in this assay?
for one of the species, an acroporid coral, which is a really important genus, it accounts for about 25% of the biodiversity of corals.
In the Pacific.
In the Pacific, and it's one of the dominant reef building or structure building species. So it's a genus that contains the species that create the habitat, what we typically think of when we picture coral reefs.
So that had antimicrobial effects.
All of them had antimicrobial effects.
And whether they're fished or not fished areas.
Yes, but this one was the only one that differed between fished and non-fished areas. And so one of the big deals now with coral reef ecology is, can you go in and do something at a local level that helps? In other words, does management make any difference? Or are all of you guys sitting out here driving to work every day, killing all the reefs in Fiji, and it doesn't make any difference what the locals do? And so what this indicated is that you're buying some insurance if you keep the reefs healthy, that they're less susceptible to these bleaching pathogens if the reef's healthy than if it's not. And the one coral genus that was really important for is the one that, if you look at the diversification of that genus through time, then coral reef fish diversify along the same timeline and stuff. It's kind of the main driver of coral structure and function for the Pacific.
So the real point is if you can do something, right?
And you can. I mean, these Fijian villagers have done something, and it makes that coral more resistant to these pathogens during times of elevated temperature.
That was another variable that you carefully compared two different temperatures and looked at the killing activity. So how are you thinking about those results?
I guess I wasn't too surprised to see. We know Vibrio coralliticus grows faster at elevated temperatures. I think the temperature optimum is around 30 degrees Celsius. So because the temperature differences are in a petri dish, in an incubator, we're looking at a single dose of that antimicrobial effect, and the pathogen is advantaged in one scenario where it's growing much faster and turning on virulence genes and things like that, probably able to degrade that single dose much quicker. But it is interesting that the beneficial effect of the protected area for acroporid is more pronounced at that elevated temperature. So it becomes more important to be on a healthy reef as the pathogen is advantaged by warming water temperatures.
So if we are concerned, as we are, about global warming, this could become a bigger issue.
It could, but I think that it's really, this beneficial effect probably has a threshold beyond which it will be overwhelmed by the warming water temperatures.
I see.
So you also looked at the microbiome of these samples?
We did.
And what did you find there?
Not a lot of differences, to our surprise. And I guess that makes sense. We were sampling apparently healthy corals, so they had no visible signs of disease or bleaching. So if you're able to withstand disease and reside in this area, you probably need to be able to regulate your microbiome to some extent. And so we didn't see these big changes in microbial community composition that I initially thought we might. We did see more nuanced differences. So for that acroporid coral, we saw that there are different indicator species. Potentially a putative beneficial microbe in the Endozoicomonaceae was more pronounced in corals, acroprid corals in the protected reefs. And then an Enterobacteriaceae, which is commonly associated with diseased corals, was more pronounced in the fished area acroprids.
And Deana, correct me if I'm wrong, but the lack of large differences in the microbiome, that's contrary to what some prior studies have shown, right?
Yeah, and I think that relates to the scale of sampling. In previous studies, researchers have, I think, sampled much more close to the interaction site. So if turf or macroalgae are in contact with coral, they'll sample right adjacent to that area or right underneath that area of contact. And so I think if you move further away from that area of contact, you might see different changes than if you're very close.
Well, and people have also sort of sampled reefs, but set it in a way that it confused it with corals sometimes. In other words, when we looked at the corals, we didn't see much difference. But if we sucked water right off the bottom of these algal-dominated versus coral-dominated reefs, we saw big differences, just like others had reported, for sort of the community in general.
Got it.
But the corals were regulating at some level.
Now, you've finished this work. You've gone on to a postdoc, right? Mm-hmm. And you're not working on coral anymore.
I'm not, though I do have some projects to wrap up still.
So I was going to ask you if you could continue to work on this coral project. What would you do?
Well, we've done some of that. We just haven't worked it all up. But I'm interested in which species of algae might be impacting coral antipathogen defense. So the fished areas, there is a lot of macroalgae there, but it might be that certain players are more important than others in terms of influencing the antipathogen defense. And I'm also interested in how corals that are experiencing warming on the reef might be influenced in terms of their ability to fight off this pathogen.
Okay. So Frank and Mark, are you continuing to study this? What are you looking at?
Well, we're doing a number of different things, but some of it is we've recently found that multiple corals planted together grow and survive better than single species, than monocultures do. And we're kind of wondering whether there's a drug cocktail that they're releasing that makes them, protects them better from some of these kind of bacterial stresses and things. One of the things we'd like to do as well is to know more about the agent that's producing these antipathogen activity. Is it the coral animal? Is it the symbiotic dinoflagellates? Is it the microbiome? And certain important species in there. As a community ecologist, we often see one or two species that make a huge difference relative to the whole community rather than big community changes. And what Deana saw is not that sort of corals in the MPA were dominated by evergreens and corals in the fished area were dominated by hardwoods, but really it was more like you've got hardwoods in all of them, and one of them you've got maybe dandelions, and they might be doing something. And so we want to go down and try to search that more actively.
All right. And what were you saying about viruses?
That could be another potential mechanism underlying some of these effects. I think it's just a super cool finding. I mean, this is essentially saying that the immune properties of the coral can be changed by what humans can do locally, right?
Just think about the fitness penalty of producing an antimicrobial to the community in general. You have to have a sufficient energy budget from the microbial ecology perspective to be able to throw that public good commodity into seawater.
Well, you may not be throwing it into seawater. You may be just throwing it into your mucus,
right?
Which is
which you're tying it down with.
Yeah, and you got to realize that corals and anything that's photosynthetic in shallow, highlight low nutrient Cs is making a lot more carbon than they can do anything with. So they're dumping carbon all the time.
So it could be a component of their secondary
Energy's
metabolism where
probably
they're just
not limited.
dumping waste energy.
Yeah.
You know, the fitness question is great, and I think it really does come down to understanding how these things are structured at the small scale. And we haven't really probed that yet. What is the spatial structure of these? And we also really haven't probed how abundant some of these agents are, right? So much of our knowledge about the microbial community is based on things that don't tell us how many actual cells are present and where they are.
Well, have you met Gina and Marvin?
Yeah, we've heard of them.
Yeah.
So one thing that I really enjoy about reading papers outside of my own field is to learn new terms that are quite widespread. Could you tell us about this Tolstoy reference?
The Anna Karenina principle.
Sure. So that came out of Becky Thurber's lab. They did an experiment down in the Florida Keys where they excluded fish from areas of reef, and they had a lot of macroalgal growth over the course of 3 years. And then they looked at the microbiomes of the corals in those areas, and they found that they, again, didn't see these shifts in composition so much as they saw a greater variability among the microbes there. And so then they went further and looked in a variety of different systems, and they see this reoccurring in humans and in mice and in different systems where if the host is stressed, that their microbiomes become more variable so that they become in a dysbiotic state in different ways.
So this is now called...
Answer the literary part.
Yeah, the literary question is...
I have the quote here.
Yeah.
And let the audience decide.
And it's from the book, the Tolstoy book.
It's literally the opening line.
Yes.
All happy families are alike. Each unhappy family is unhappy in its own way.
Yeah. There you go.
Anna Karenina.
Yeah.
And if you haven't read it, you have to go read that as well.
It's really a remarkable concept, and if you think about it, it sort of ties the two papers together. It really does. If you sit and listen and go read her PNAS paper, which is open access, and read this one, which I assume yours is going to be open access.
It is.
Read them and think about it when you read this reference from Tolstoy.
I think it's also useful. Gina and I talked about this after reading their paper, and we were thinking about not thinking about it in terms of the bugs, but the functions that are there, right?
Yeah,
it needs
absolutely.
to be a functional outcome and not a bug outcome. Right?
Yes.
And I think it's the way everyone's beginning to interpret microbiomes. And I think, and what you see in the oral cavity at least is that things are not always different in different ways. If you have a dysbiotic community, there are functions that are always maintained no matter who's there. And so in those cases, I don't think this applies. In other cases, you do see functional differences in all of these different functions, but there are uniquely conserved functions for a disease community, I think. And butyrate production is one of those, I think, in the mouth.
So you could look at the virome because you still have these samples, right?
We do.
You do.
Yeah.
You could sequence and see what's present because people have done that, and I know herpes viruses are a major component of coral, but there are probably others as well. And in your case, you have these two environments to compare. Would be interesting to see that.
So let me ask, since I didn't read the methods in detail, did you use NGS to get your 16S data, or did you just do straight 16S?
We did aluminum MySeq sequence.
So you just threw in the adaptomers and you amplified up the entire
Community.
genome present. So you could answer the virome question
No, we went in and targeted the 16S.
Okay.
But we have the samples that would enable us to do a truly metagenomics approach.
So if you go back and then you just have one of these aspiring young scientists in our room do a genome
Marvin
hunt.
could do it for us.
Yeah, that's right.
Marvin can do it for
He's
us.
got nothing else to do.
We just volunteered you for something.
Gina needs another project anyway.
Gina needs another project.
So we're the guests of your center, Microbial Dynamics and Infection. These papers really illustrate so well what you all are doing here. Do you want to talk about the birth of this center and how this illustrates your vision?
Sure, I'll start a little bit and then let these guys. I mean, I think, the center started a little over a year ago, maybe a year and a half ago. We got off the ground, and the basic idea has been that we had a lot of microbiology here at Georgia Tech, and it was sort of disparate, I think, a little bit. It was in maybe 4 or 5 buildings, and a lot of people doing really interesting things that you can actually draw parallels to, but not really, I think, a community that was maybe as together as it should be. And the university has put forth a lot of resources to actually refurbish a building that most of us are going to actually be in. Now, a lot of us are already in that building, and I think it's made a big difference where the students can actually see each other. I think we talked about it earlier today. I think the trainees are where these interactions are actually really important. The faculty, you fly to Fiji, right, and you hang out, right, and these sort of things. We don't see each other all the time, but it's the students and I think postdocs that see each other. And so getting us into the same building together, and then some resources for us to start helping with the training environment and do things like professional development and those kinds of things. But it's really asking about scalable principles, right? I mean, can you learn things in an abscess model that you can actually use to think about things in these more complex systems, natural systems like corals? And I think there probably are some of these principles. And then we have a lot of really interesting, we're kind of different from the other microbiome centers. We have physicists and math people and
Evolutionary biologists.
And I think so. I guess the way that we sort of approach questions or thinking about them in maybe a little bit different manner than just asking, who's there, but asking questions about who's there, but where are they, right? If they're here, do they do one function? If they move over here, do they do a different function? And I think that's the real key aspects that I think we're going after. I don't know, Frank, what you think?
No, I think all of that is true. We spent a lot of time at the inception thinking about what differentiates us from other folks that have tried to do something similar. And we looked across what the faculty are doing here and what we can be good at. And, and it came down to much of what is going on in our labs has to do with how microbes interact with one another, right? And so this idea of microbial interactions was something that we really wanted to elevate as a core theme. And we wanted to think about what that means, both in terms of spatial interactions and interactions in a unit of time, but also across time, right? And so that could be evolutionary time, it could be something over shorter time scales, and what were the terms that could kind of capture that? And microbial dynamics is kind of where we ended up with that, and we recognize the way that term can extrapolate across systems. I mean, we're working in very disparate systems, but in many ways, some of the core questions are the same, right? And then we also wanted to make sure and that the message got out that many of the questions that we do ask actually do have translational applications, or at least the understanding that we can get from them has translational applications. And much of that is related to how we understand things like infection. So that had to be an important component of how we define the center as well.
And I think, we really want to be quantitative, I think, and that's the reason I think that some of the value of being at Georgia Tech is the quantitative aspects. We don't want to know just where you are. We want to actually know sort of build models of emergent properties. Why are you there, right? How did you get there? How did the spatial structure form? This requires modeling and thinking about things more quantitatively and just image analysis to modeling, I think.
It's so interesting to me that the questions that you're defining for your science also apply to what you're trying to build as a center.
Right.
Interested in the interactions between different groups and how they can benefit. And
yeah. The bugs are easier.
Yeah.
Deana,
They work faster.
They do.
I wanted to ask you, so you're at a postdoc now. What are you doing for your postdoc?
I'm working within a collaborative group, and Drew Harvell, Emmett Duffy, and Jay Stakowitz, they have an NSF to investigate eelgrass wasting disease.
Eelgrass.
Eelgrass, so seagrass.
Seagrass.
And up and down the California coast, all the way up to Alaska. And so we're sampling to look at prevalence of disease and trying to understand drivers of pathogen. It's a protist that causes lesions and die-off events in some cases.
So a different system, but similar questions.
Similar questions.
That you've been trained to be able to address, right?
Hopefully so.
Okay.
Yeah.
And beyond that,
You
what
need
do you,
a thicker wetsuit.
Yes.
And maybe
Yeah.
snowshoes.
Yeah.
it's colder, right?
Yes,
The
much
water's
colder.
Yeah.
And muddier, but it's pretty fun and beautiful there.
Okay. And beyond that, what do you think of the future? What do you want to do?
I'd love to continue doing research. I'm a bit more open-ended in terms of whether that's faculty position or a research institute, NGO. And mentoring students has been really rewarding experience, so I'd like to continue to do that.
Okay.
I'm curious whether Gina and Deana have any advice they would like to share with other trainees as they reflect on their own paths.
You can go first.
I was going to say 25 is probably too many.
Some of the best advice I got when I was going into grad school was that the PhD is a marathon, so pace yourself. Always take time to invest in the things that you enjoy outside of work, and that it's very common and normal to doubt yourself through the process, but if you think that you're not capable or not smart enough or you don't belong, you're wrong. So,
Good advice.
I think the advice that I was thinking about is kind of along the same lines, but sometimes your experiments work and that's really exciting, but a lot of days they don't. And that's okay, and that means you're normal. And I think the satisfaction of like getting them to work eventually is really strong. And so, don't get too discouraged and keep working through things and think creatively about how to make things successful in the end.
It's why we call it research and not search.
cute.
Marvin, if you hadn't been a scientist, what would you have done with your life?
What would I have done with my life? I'm 5'7", but I would like to have been a basketball player. I don't know what I would do. I mean, I lived in a place where no one went to college. So, all my buddies are back there selling cars and doing those kind of things. I mean, I can't imagine not doing science. I mean, from when I was a kid, I saw a tornado. I guess I didn't get to say, but the reason I got into science is I saw a tornado when I was in Texas, and I started getting all these tornado books from the library and asking how this works. And so I don't know, I guess.
Hence the aerospace.
Yeah. And so I guess I've just never thought about it, to be honest, about what else I would do. I mean.
Okay. Gina, what do you
My
think?
turn.
What would you're still early on, but you can still answer it.
I don't know. I never felt like being a scientist was my default, I guess. I always liked science, but I always liked a lot of other things too. I've always really loved history, and I could have seen myself, majoring in history in college instead of biology and then going off and doing, who knows, I don't know what you do with a history major, but,
You're going to get letters from history department. be aware. They do write. They do write.
They write books, yeah. They teach and write books, yeah.
Yeah, so I've always loved kind of teaching, even with my peers in school growing up, so I wouldn't have been surprised if that's in some aspect where I ended up, no matter kind of what field I was in.
Okay. How about you, Deana?
Do the hosts not have to answer?
Them, Michael, and did I ever ask you that question?
I have an answer. I think I would coach women's athletics.
How about you, Michael?
I'd probably have been an engineer. From my earliest childhood memories, I would be I took things apart and tried to put them back together, and it's probably why I'm a scientist. I want to learn how stuff works.
Actually, I think you'd be a good talk show host.
Yeah. There's more money in it than science. There's more money than science.
Deana, that was a good idea. Thank you.
I guess while I have no musical talents, I'd love to be a musician. I enjoy listening to music. If I could just pick and magically have those skills. But no, I can't say that I've really thought too much about it. I've always really enjoyed doing science. So.
Well, that's why it's a fun question, because we'd like you to be spontaneous. A lot of scientists answer music.
Really?
Yeah, it's interesting. Yeah. Mark, about you? I can answer for you, but I'll let you answer.
Well, my only alternative skills were construction and lifeguarding, neither of which I really wanted to continue doing. And so I don't think I'd do anything else. I think, my job is to find out cool stuff and tell people about it. I can't imagine anything that's better than that, I don't know.
Okay, that's a good answer.
Yeah.
How about you, Frank?
Well, my 8th grade teacher told me I should be a priest.
You went to Catholic school?
I did, yes. 4 years later, I doubt if she would have said the same thing. But,
Nobody that knows me's ever suggested that.
It's a good question. I've always enjoyed being on my own in the mountains with a camera, so I can envision doing something along the lines of photography. I also really like communicating science in the written word, so I bet I could have fun with that as well.
Okay.
Vincent, now you have to answer your own question.
Oh, I always wanted to be a scientist. Nothing else.
Oh, that's a cop-out.
No, it's not a cop.
At what age?
Just out of
Out of the womb?
Out of the womb. No, I can't remember back so far, but I loved tornadoes. I loved stuff in the backyard.
Yeah.
And then my dad was a doc, so I used to think medicine was science. But now I know it's not. But that got
You're
me.
going to get letters for that one.
That's why, as Alan Dove said, we have some docs, MDs out here. No, I'm not going to offend anyone. Good. Alan Dove says, I love this. MD is a training program, PhD is an education. And that is so true, right? It's so true. One more question. RNA or DNA?
RNA.
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 DNA.
RNA.
I don't know what.
BMW, they make faster motorcycles.
Do you know the difference?
Yeah.
Oh, I get it now.
Yeah.
DNA, of course.
Okay. That is TWiM 208. If you listen on a podcast player, we'd like you to subscribe. If you don't listen, please subscribe. We need to get more listeners. It really helps us able to raise money. Show notes are at microbe.tv/TWiM. Questions and comments, twim@microbe.tv. As I said earlier, if you like what we do, consider supporting us. Go to microbe.tv/contribute. You can use PayPal or Patreon and give us as little as $1 a month. It's nothing for you, really. It's less than a cup of coffee. Even here in Georgia, it's less than a cup of coffee. And it will really help us because there's strength in numbers. Microbe.tv/contribute. Our guests today from Georgia Tech, Frank Stewart, thank you so much.
Thank you. It was an absolute pleasure.
Mark Hay, thanks for joining us.
Thanks.
Deana Beatty, thank you. Beatty.
Yep.
And good luck.
Thank you.
Gina Lewin,
Thank you. This was fun.
and good luck as well. And Marvin Whiteley.
Thank you, and thank you guys for coming. I think we should give them a round of applause.
And of course, Michelle Swanson, she's at the University of Michigan. Thanks, Michelle.
Thank you, and what an impressive collection of scholars you have here. It's really would be a great place to work and train.
Michael Schmitz at the Medical University of South Carolina. Thanks, Michael.
Thanks, everyone. And I want to remind everyone in the audience that you're our editorial board. So if you happen to stumble across a paper you find compelling and think other people on TWiM might want to listen to, drop us a note and we'll be happy to review the paper and, solicit comments from the greater world.
I'm Vincent Draccaniello. You can find me at virology.ws. I want to thank Brandy Ulmer for logistics. Thank you so much for helping. I want to thank ASM for their support and Ronald Jenkins for the music. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology. Thank you.